remember that we have our duodenum, and then we have the chyme that's very acidic. And now we have our pancreatic duct cells that are producing and the acini that are going to produce pancreatic juices. So remember that our chyme from the stomach is very acidic. You have all this hydrochloric acid. But what prevents your chyme from digesting your duodenum and your pancreas? Well, the pancreatic duct cells are going to produce a chemical called bicarbonate. And this is the same bicarbonate we talked about when we talked in the lectures from the respiratory system. So especially for exam 3, now I definitely want you to know the bicarbonate buffer system because it's the final section. We have to talk about it. So bicarbonate is a weak base. So what's that going to do is react with the protons from the hydrochloric acid. And remember that the bicarbonate buffer system is a reversible reaction. You can take carbon dioxide and water and then make bicarbonate and protons from that, but you can also do the reverse. You can take bicarbonate, react with the proton, make carbonic acid, which can be broken down into carbon dioxide and water. So that's what happens here. So what happened is that we used up bicarbonate, but we also used up a lot of protons by remember that the basis of pH is proton concentration. What we did is neutralize a bunch of protons. Now we have carbon dioxide and water, which in that form doesn't affect pH. Now, if carbon dioxide started making this again, yeah, it would affect pH. But in this state, the pH is now neutralized. And then, how about the duodenal epithelium? Well, the thing is that the duodenal epithelial cells also produce a lot of bicarbonate as well. So not only do you have mucus that protects your duodenum, the duodenum and also secretes its own bicarbonate that mixes in with the mucus. So now you have a physical barrier against the chyme, the acid from chyme, but you also have a chemical barrier that helps to neutralize any acid that somehow tries to make its way past the mucus layer. And that same reaction is going to happen. Bicarbonate is going to react with hydrogen or protons to form carbon dioxide and water. So this is why acid-base balance is kind of important in your gastrointestinal tract. And then what we have here is saliva. Again, I'm not really concerned with you memorizing these specific numbers right here, but you should know, like, compared to, it's not quite neutral, it's not quite basic, but it is toward the neutral-ish pH range. Now, gastric acid, way more acidic than saliva. And then the duodenum is not basic or alkaline pH, but it's relatively more basic than the gastric acid in your stomach. So what we have here, why is the gastric acid so acidic? Because of the hydrochloric acid. It's a very strong acid. But in the saliva and duodenum, what you have are bicarbonate buffers so you're going to secrete pancreatic and duodenal bicarbonate in the intestines and pancreas. But you also have bicarbonate being produced in saliva as well. So what happens is that you have kind of a chemical barrier that prevents your gastric acid from dissolving your esophagus and also prevents your gastric acid from dissolving your intestines and other organs downstream of the stomach. So this is why acid-base balance is kind of important, because not only do you have those sphincters that kind of try to contain stomach acid, you also have a chemical barrier at from both saliva and the duodenum, trying to keep gastric acid where it's supposed to be. Now you also have a host of enzymes all throughout your GI tract. So salivary enzymes, like salivary amylases and lipases. The salivary brand of amylases and lipases will work best at the saliva pH. So they work best at this kind of like slightly acidic, just below neutral pH. But gastric enzymes, remember that gastric acid very acidic due to hydrochloric acid. So pepsin and like gastric lipase, they're going to work best at these very acidic pHs. Now, duodenal and pancreatic enzymes, well, they're not going to function at very acidic pHs. They're going to function better at the natural pH of the duodenum. So like pancreatic amylases, lipases, proteases, nucleases, they're going to work best at pH 6 to 7. So again, more compared to gastric acid, relatively basic in the mouth and duodenum. But yeah, so the enzymes work best at the pH that they're being secreted at. So say we have a salivary enzyme, and then it's being washed down via the bolus and saliva into the stomach. Well, what's going to happen once it enters that very acidic pH is that this enzyme is going to denature. The structure of this enzyme is going to fall apart and thereby be inactive. And they can also be chopped up by proteases as well. Now, gastric proteases like pepsin, they're going to function best at their acidic pH, but what happens when they move to a more basic, relatively basic pH? Well, they're going to denature, they're going to become inactivated there. And what's going to function best at that pH at the duodenum? Well, the enzymes that are secreted there. So things like pancreatic enzymes or small intestinal enzymes, they're going to function best at this pH range.